Welcome to the class of High Throughput Data Generation and Analysis. Dear students, in the today's class we are going to learn about how we should analyze the gene expression data and what are the different methods. So after the normalization of the data, as we have seen in the last class, that we should normalize the data after the transformation, the normalization is conducted. So this data, then after the processing of the data is then presented in the form of matrix, that is called expression matrix. So each row in the matrix correspond to a particular gene and each column could either correspond to an experimental conditions in the expression matrix. So in the column we take the conditions and in the row each row denote a particular gene. Expression level for a gene across different experimental conditions are cumulatively called the gene expression profile. We can call this gene expression profile because for the same gene, for a single gene, if we want to see the expression of the same gene in the different conditions, that we can see across the row. I mean in the single row we can see, because single row contain the different columns, different intensity from the different conditions, the data from the different experimental condition like aerobic, anaerobic or pH 1, 2, 3, 4. So in each row we have the different column, means the expression of the same gene and different conditions can be analyzed. So this gene expression profile contain that. In the gene expression profile we have the expression of gene in the different conditions. Okay, so that is called gene expression profile. And in the sample expression profile, what we call means for the column, for the single column we can say this is the sample expression profile because that belongs to the same sample, but it contains all the genes that has been expressed from that sample. It doesn't contain the different environmental condition. So single column denote the single experimental conditions. So expression of all the genes within the same condition that belong to the same sample. That is called sample expression profile. Now, so sample expression profile we can also see here like this. So this you can say this is the sample expression profile. Here you can see expression profile of a gene I can be represented as a row vector. So here you can see x11 means first row first column, x12 means first row second column, x1n means first one and column. So it will go up to the and column. So here in the second row also you can see second row first column, x22 means second row second column, then x2 and means second row and, so it will go up to the and columns. So this is called, what this called means we will show this gene expression profile. This is called gene expression profile. An expression profile of a gene I can be represented as a row vector. Gene I and gene I1, gene I2, gene I3 means this is the row, first column, second column, third column. IN means I means row. So it will go up to the and column. So this is known as row vector. So we will call this gene expression profile, GI. Okay, now it is clearly understood. So expression of the same gene within the different, under the different environmental condition, different experimental conditions. Like gene, the same gene can be, we can analyze the, we can take the sample, the similar gene and the different, we can allow the expression of the gene in the different condition like aerobic and anaerobic. So that will have the two column aerobic and anaerobic conditions. So the gene can be expressed in the aerobic conditions also and in the anaerobic conditions also. Some of the gene can be expressed in both of the conditions and some of the genes will be expressed in only in the one condition and some of the gene will only be expressed in the anaerobic condition. So this will be shown here by the gene expression profile. Okay, so that is denoted by the row vector. Now come to the expression profile of sample. Sample J can be represented as a column vector. So here you can see Gi means Xij, X2j, Xmj, means it shows the number of the columns like J1, J2, J3. Okay. So X1, X2. So because see, within the same column there is no change in the experimental condition, so it will be shown by the column vector. So this is known as sample expression profile. Okay, so this is all about the gene expression profile. Okay, so for analysis of the expression data, then we can analyze the supervised learning and unsupervised learning methods. Okay. So this is about the representation of expression profile. Okay, now you have understood what is the expression profile. So in the expression profile can be denoted by the two vectors, row vector and column vectors. So after processing of the data, genes expression data can be expressed as in the form of expression matrix. Okay, so in which we have column vector and row vector. So expression profile of a gene can be considered as a vector in n-dimensional space where n is the number of conditions, and an expression profile of a sample with m genes can be considered as a vector in m-dimensional space where m is the number of genes. So because m shows the rows in the m means when we have means number of rows and number of columns. So m is the number of rows like m1, m2, m3. So this will show the different number of rows within the same column. Okay, and is the number of the conditions means n1, n2, n3. So this will contain the single row and multiple columns. Now gene expression profile is clear. So gene expression matrix acts with m genes across n conditions is considered to be an m into n matrix where the expression value for gene I in condition J is denoted as Xij. So here it is clear. In the expression matrix, okay, we have rows and columns. Okay, so number of genes are shown across the rows. Means you can see the on the vertically or and you can you can count it here like x1, x2. So these are the number of rows. Okay, number of rows x1, x2, x3. Okay, now column is the same here, 1, the first column. So column shows the conditions and the row shows the number of genes. So column shows the experimental condition. Now it is clear. So expression profile will be shown by the Xij. Okay, so this is the matrix. So in this we can see this is the row vector and column vector. So now the expression profile is clear. Okay, now this topic is over.